Let me introduce myself. We have some people who are new to us. My name is David Bland, and I've been a member of Ethical Culture for almost 20 years, and I'm also the administrative director of the society, which mainly means that I pay the bills and do a lot of the clerical work. Prior to that, I spent about 35 years in IT in the financial services industry. I'm really happy and honored to welcome Brianne Barker, PhD, who is an assistant professor of biology at Drew University. Dr. Barker earned her PhD in immunology at Harvard University and is also co-host of the podcast This Week in Virology, which is where I learned about her. It's also known as TWIV, and it rocketed to worldwide fame, literally, when COVID hit. Dr. Barker has agreed to talk to us about all things COVID, and I would also like to take the last part of this interview to talk about women in the sciences and the obstacles they face, which is a topic I know that Dr. Barker is passionate about. So here's how I would like to work this. I'm going to interview Dr. Barker first on the topic of COVID, and I have a bunch of questions to ask. So I will ask you all to hold your questions until I'm done, when we will open it up for a general Q&A, and it is my intent to leave plenty of time for that. But if you have a burning question that you just can't stand keeping quiet about, please put it in the chat window during the discussion, and I'll try and monitor that and get to it. So with that said, thank you very much, Dr. Barker, for giving up part of your Sunday afternoon to be with us. And I'd like to start with some basics. First, the world is talking about Omicron. It seems like nothing but Omicron. So the Omicron variant. So what is a variant? What is the Omicron variant? And how do variants occur?
So variants are different versions of the virus that have acquired some changes in them compared to the original virus. Viruses are mutating all the time. The virus that causes COVID, SARS-CoV-2, can be a little bit messy in its copying process, so that it makes some mistakes when it makes copies of its genetic material. And that's how we get mutations and we get different versions of the virus. Sometimes you will get sort of a new version that seems to do well and reproduce a lot. When we see one variation or variant take over, then we might study its properties a bit and decide whether we might call it something called a variant of concern. And once it's a variant of concern, it gets a fancy name and it gets even more study. And so Omicron is a recent variant of concern. It was detected around Thanksgiving, and it has something like 50 changes compared to the original version of the virus.
And we've all heard a lot about the spike protein. What is that?
I feel a little bad because when I'm in my office, I have a model. I don't have my model here, but you can imagine the virus.
I have a picture.
Okay. You can imagine the virus sort of looking like a sphere and having sort of little lollipops off of the side coming off of it. And you can see that quite nicely here. You've got the virus as a whole as this nice sphere, and you've got these red projections coming off. Those red projections are the spike protein. They are what the virus uses to interact with a cell, sort of like a key to get into the cell's lock. And they also are an important target of the immune system, since you can see they're so big and obvious on the outside part of that virus.
And when people talk about these mutations that have caused the Omicron variant, where is that on the virus? Is that in the spike protein that they're talking about?
So there are about 50 changes in the spike protein. about 50 changes in the virus, about 30 of which are in the spike protein in Omicron.
And we'll get more into this. I'm going to close that picture now, but we'll get more into this. But so how do the vaccines, what do they target?
All right. On the left, you can see a version of that virus that you saw before. Now it's a cross section, so you can see some of the inside. Your immune system is able to target that virus in a few different ways. The vaccine only includes the information for the spike protein. So you end up making immune responses against the spike protein and not the other portions of the virus. That is important in terms of how that is able to work. The spike protein has lots of little areas in it. It's shown here as being pretty small, but in fact, it's a huge protein in the overall size of protein. So you can see I've drawn 3 little red dots to show you some parts of the spike protein. Your immune system will make some antibodies that recognize spike, and will also make some T cells that recognize spike. And those antibodies will recognize one of those little patches on spike, not necessarily the whole thing. So in fact, you might have 100 different types of antibodies that are responding to 100 different types of targets in spike. You also will have little patches that are recognized by T cells to help get rid of that virus. So we've got some antibodies and some T cells that are recognizing little patches in spike.
Now, we've heard a lot of press reports about Omicron being more transmissible, but less virulent. So number one, do we know if that's really true? And what does it mean when we say it causes a less serious illness?
So from all of the data that we've seen, it does seem as though Omicron is much more transmissible. The virus does seem to move between people quite easily. But there are lots of things that could cause that in terms of how the virus works. And we don't know exactly why it's more transmissible yet. The question as to whether or not the virus is more or less severe is much harder to answer. The data that has indicated that the virus is less severe comes from the idea that there are a smaller percentage of people who are going to the hospital with this virus. And that was particularly true in South Africa. Unfortunately, if you look at the data in South Africa, or I guess fortunately, because they didn't have very severe disease, those individuals who were getting infected in South Africa were much younger than people who had been infected before, because most of the people who were older had been vaccinated. Most of the people who were getting infected with Omicron, in some cases, might have some immunity from previous infection or from vaccination. And in the past, people didn't have immunity. And so things like those changes in the people who have been infected this time around, in their age, in their other disease states, and in their immunity, make it sort of an apples to oranges comparison to say whether this is actually a more severe virus or not. And I think people are still kind of debating that one. Actually, one of my podcast colleagues I was call these things apples to oranges comparisons. He refers to it as apples to wrenches. And that's, I think, a more appropriate consideration here. They're just two very different populations when we look at the number of people who are in the hospital now versus the number who were in before. So I will say, yes, it is absolutely more transmissible. I don't think we know really well whether it is more or less virulent.
Okay, and I see we have a lot of questions coming in the chat window, and I will get to them, but I want to get through a few more of my own. So there's a lot of confusion I've noticed over the mRNA vaccines, how they work and how they differ from traditional vaccines, as well as from the J&J vaccine. So could you just talk a little bit about that? And I've heard many people ask one specific question. Do the mRNA vaccines in any sense alter someone's DNA?
Okay, great. So mRNA vaccines, well, so actually, I guess I'll go into sort of the more classic versions of the vaccines. So many of the vaccines you've received in your life in the past have involved either a version of a virus that has been weakened in some way, or a version that has been killed. And that gets tricky with virology about viruses being alive or not, but we'll just say killed and go with that. And so basically either it's a weakened version that can still reproduce, or a sort of dead version that can't reproduce. And it's the whole virus. The good parts of that are that your immune system can sort of see the whole virus, but it might have a little bit of risk associated with it. And what we've realized from doing many, many years of immunology studies is that your body really only needs to see one great target. It doesn't need to see the whole virus. And that spike protein in other coronaviruses has been an amazing target. And so we are making mRNA vaccines that just include the instructions for your body to make that one amazing spike protein target for a brief period of time, and then allow your body to know how to recognize that spike protein moving forward. And so we're using something called messenger RNA technology. Messenger RNA is in all of your cells right now. You have messenger RNA for insulin in the cells in your pancreas that make insulin. It's part of the normal process of making different proteins in your cells. So your cells right now are full of mRNA. It's sort of a short-term messenger. That's where the M is from in the name, that carries a message around your cells. And so we've made messenger RNA to tell your cells how to make the spike protein. We put a little bit of it in your arm, and only the cells that get the injection in your arm get that message. They make the spike protein and they teach your immune system, which will then go through your body looking for the spike protein. So you only have a small number of cells in your arm that are getting that mRNA and making the message about how to make this target spike. mRNA does not have the ability to interact with your DNA. It's in a different part of the cell. And it's chemically different enough so that the two are not going to, the mRNA is not going to be able to insert into the DNA. The nice part about an mRNA vaccine is that we can make it quickly, of course, but also we don't have to deliver all of the parts of the virus. So some parts other than that good target spike, which may be important in causing disease, aren't there. So we don't have to worry about that leading to you potentially getting sick. And we also have the perk of the mRNA giving us a very strong immune response. Sometimes people are worried about mRNA vaccines in their fact that they've been generated so quickly. But I will tell you that when I was in graduate school in 2002, I was working on vaccines like mRNA vaccines. And there were mRNA vaccines that actually went into humans to act against rabies in 2013. They just didn't have a lot of money behind them, so they didn't go very quickly, and that they actually didn't make it to market because they didn't have all that money behind them. So there have been mRNA vaccines in people since 2013. They've been in research for much longer than that. It's just that they kind of hit a home run this time, and we were very lucky.
Well, there's certainly a lot more money in it now, right?
A lot more money. Lot more money.
So do the mRNA vaccines, or for that matter, the Johnson and Johnson vaccine, do they give you a little case of COVID?
They don't, because you are just having the one part of the virus, the spike being made in your cells. You aren't having the rest of it. With the Johnson and Johnson, basically we're giving you the information to make that spike message in a kind of shell that's derived from a different kind of virus. It's really very similar in terms of the concept. What I will tell you is that sometimes you do have some side effects. Sometimes you have a bit of a fever, a bit of aches, something like that. That's not a case of COVID. It turns out that many of those symptoms are symptoms that happen when your immune system gets turned on. So when your immune system gets turned on, you start to have a little bit of a fever. And that's actually because your body's trying to change its metabolism and how it deals with sugars and things so that it can make a better immune response. Your body is doing a few different types of changes when you make it an immune response, and that gives you some symptoms. And so you might feel some symptoms when you get a vaccine. That doesn't mean that you're getting COVID. It's not necessarily pleasant. I don't enjoy it either, but it is a sign that your immune system is doing its job.
So you said that those symptoms, which many people are quite familiar with, are because your immune system is going into action. So is that why many infections produce the same symptoms, right? And you get sick and it creates a fever. It seems like no matter what you're sick from, you get the chills, you get redness, you get blah, blah, blah. Is that the reason?
Those non-specific symptoms are all related to the immune system, yes.
So it's because these diseases all trigger the immune system.
They're all triggering the same part of the immune response, yes.
Okay. Now, this is a good time to talk a little bit more about the immune system, which I thought I knew a little bit about when I started listening to TWIV, and it turned out I knew nothing about. It is the most amazing and complicated system I've ever heard, and I'd estimate I understand about 10% when you guys really get into the nitty-gritty, if that. So could you talk a little bit about innate versus adaptive immunity, T cells, B cells, memory B cells? How does all that stuff fit together? What are neutralizing antibodies? What are non-neutralizing antibodies? Blah, blah, blah. If you could do that.
Sure. Sure. So your immune system, I often think about as having sort of different layers. And those layers are kind of the levels of protection or the walls of protection. And they will function at different times, and they will sort of protect us in sort of different ways. So the innate immune system is a part of your immune system that is acting really early following infection. The adaptive immune system takes a little while, maybe takes up to a week to get ready. So your innate immune system is the thing that's acting early. The adaptive immune system is acting a little later. The innate immune system is also a little bit less specific. The adaptive immune system is the thing that's really going to get to a particular, this year's flu versus last year's flu, or particular versions of COVID. We mentioned before that there are these symptoms that are coming from the immune system that are similar between different viruses. That's because they're all coming from the innate immune response, which is similar with different responses. Within innate or adaptive immune responses, you can have different types of cells and molecules acting. The two big stars of the adaptive immune system, so this is the specific and later part of the immune system, are called T cells and antibodies. Antibodies, everyone likes to think about antibodies as being little superheroes, but actually they're just little proteins that are made by different cells, particularly a type of cell called a B cell that can look like a little Y shape. I drew them as Y shapes before, and act against viruses T cells are cells that could kill infected cells. And so we've got an ability to disable the virus before it gets into our cells, and an ability to kill cells once they are infected. And so we have those two different arms of protection, and we always use both of them in helping us act against a particular microbe. Again, those antibodies are coming from a special kind of cell called a B cell. Again, with the immune system, we like to divide things up. And so we can divide up, the B cells and the T cells into some that act right away and some that actually live throughout your life to protect you in an improved way if you are infected again. And so those are called memory cells. They are sort of better equipped to help you out a second time around. There are others that become really good antibody producers that aren't necessarily supposed to live for a very long time. So we can divide cells up that way. We can also divide up the functions of some of these molecules. So neutralizing antibodies are one kind of antibody, and neutralizing antibodies work by physically getting in the way so that the virus cannot interact with our cells and can't get into our cells. Those are the types of antibodies people look at most often in the laboratory. When we are looking at vaccine responses, they may or may not be the most important ones to be looking at here, but they're the ones that people have always looked at all the time.
And now we have a question in the chat window, which is very appropriate here. A couple of people have asked, how come you can get COVID even if you've been vaccinated?
Great question. So I am actually going to show you a slide here. This slide always sort of gives me an interesting feeling to talk about. Because this is a slide that I actually made. This slide and the next slide that I'll show you are slides I made in 2009 when I was in graduate school. And I was making them to talk about a particular piece of immunology, and I never realized that they were going to be some of the most important slides that I would use over and over again when talking about COVID. So this is just showing you some general ideas of what an adaptive immune response often looks like. You've got the size of the response on the y-axis and time on the x-axis. And what you can see happen is someone get infected and of the virus reproducing in them. You can see how long the virus reproduces here, although this doesn't show you how much virus you have. And you can see that the immune response takes a little while. There are a few days where that immune response isn't doing too much. But then our immune response actually comes up to a nice high level. And when we get that immune response at a nice high level, that stops the virus is gone. You can see also when the virus is in your body for a little bit of time, that makes you feel sick. You've gotten virus maybe to reproduce to a high level. You feel sick for a while. When the virus is gone, you start to feel better. Now, most people know that, for example, you may not be able to get the chickenpox twice, or there are certain infections that you don't get twice. But in fact, what will really happen is you will get infected a second time. And you can see that being shown here with the arrow. And the virus will get in your body. It will colonize you. It will reproduce a little bit for a short period of time. You can see it's a much shorter period of time here than it was before. And if I was showing you the amount, it would be way less. In fact, it's so little, you never feel sick. You don't even know that you are infected. Your immune system knows, because your immune system makes this big, giant response. And that big, giant response gets rid of that virus. It sort of nips it in the bud so that the virus is gone and you don't ever feel sick. And if I said to you, did you get the chickenpox a second time? You would say, oh no, definitely not. I felt great. And so what you should realize is that just sort of the basic idea of how the immune response works is that it will allow you to potentially get infected a second time, but it will act sort of like a fire extinguisher and put that get rid of that infection very quickly before it leads to any kind of illness, and before you lead to problems, of any sort of problems. And so what we're doing with a vaccine is we're giving you some kind of preparation so you can make that first immune response. So if you ever encounter the virus, you will get that same kind of infection and that same excellent second immune response, but no disease. And again, I made this in 2009. And the idea there was that you could get an infection after vaccination, but you wouldn't get disease. You get an infection for a day, your immune system would clear it up and you'd be fine. You wouldn't know that you were sick. And that's the way that sort of we've always thought of the immune system as working. The difference now is that before I was asking you, did you get the chicken pox? And you were saying, oh no, definitely not. Now we're doing very sensitive tests to look and see if that virus got into your body. And we're saying, oh yes, in fact, it did, as we might have expected. And if we had been going back in the past to look and see, does the chickenpox virus get into your body a second time, we would have found out that it did as well. So part of the reason we're seeing people testing positive is that we're using a very sensitive test to look for the virus. We didn't look for the virus before in other cases. And so that's what's going on. And you also can see in those curves kind of what the immune response should look like. Most importantly, that it should go up and down. If it didn't go down, you would turn into one big walking lymph node pretty quickly, and you'd never have enough room to make T cells to respond to next year's flu because you'd be full of T cells for this year's flu. So it's very important that you have that response going up and down as well.
Okay, so I'm going to ask one other question about rapid tests, and then we've got a lot of people with a lot of questions. I'll try and go through what's been in the chat room, and then we'll open it up. Okay, so been a lot of interest about these rapid antigen tests, which you can use at home. I've used them myself. I have 3 boxes of them upstairs. So what are they used for, and how do they work, and are they the secret weapon to getting us through this crisis?
So the rapid antigen tests are testing to see whether or not you have the virus in your body, particularly in your nose, usually when you're doing a nasal swab. They differ from some of the other types of tests. So you also may have heard of a PCR test. The PCR test is also looking for virus in your body, but it's looking for the specifically for the genome, and it is very, very, very sensitive. It can find even very tiny amounts of the virus. The rapid antigen test, like the one I have over there, is looking for another part of the virus, another protein, and it's not quite as sensitive. It's not going to find very, very, very trace amounts. It turns out that that rapid test, however, will tell you if you have enough virus in your nose that you could spread it to someone else. Sometimes you might have a little bitty trace amount left over, say if you're getting over an infection, and that might be too little to spread to someone else. But if you have enough to spread to someone else, that's enough to be positive on one of these rapid antigen tests. And so it is sort of looking at something slightly different than you would have with a PCR test, which is looking if you have absolutely any of the virus in you at all. But if you're worried about, say, going to visit family members, I took a rapid antigen test on Christmas morning before going to visit some family members in order to make sure that I didn't bring the virus to them. That might be a good sort of idea. And so that's what those tests are going to be used for. And they can be very helpful in helping us know if we're going to expose others around us.
Okay, I have 2 more questions about the rapid tests.
Okay.
One is, I've had at least 3 people tell me this. I went to a family party and we all took rapid tests that morning and everyone was negative. We met on a Saturday, and on Monday, so-and-so came down with COVID. What's your reaction to that?
My reaction to that is that is certainly something that's possible. There's time between your party and the positive test. You may have done something else in your life in between the two. And it's also, you could be very unlucky that maybe you took the test in the morning and you had a low amount of virus, not enough to test positive. And then when you went to the party, the virus had reproduced enough that you had enough to spread. There are a few different ways that can happen. And so rapid tests are not going to be sort of a perfect panacea here. But they are certainly going to help someone know, oh, I'm positive, I need to not interact with others.
Okay, one more question on the rapid tests. So at our group, Ethical Culture, we have a policy. We've stopped meeting in person for January because it's spreading like a wildfire. But we have a policy that everyone has to be vaccinated and everyone has to be masked. And there are a couple of people who are not vaccinated for their own reasons, and they have asked, why can't I just take a rapid antigen test? And then if I'm negative, participate. So my question to you is that safe? Is that a safe approach?
I think that that is an approach that is really a question that your community has to come up with in terms of how you feel about the comfort level. You can think about whether people are wearing masks, which, as you mentioned, they are, and you can also think about the risk level of different members of your community. I personally would tend to spend a lot of time around mostly vaccinated individuals, and that's because I know of some people in my life who are immunocompromised. And so I really try very hard to not to do anything that could have a low risk of acquiring the virus, because I don't want to spread the virus to my immunocompromised people in my life. And so since I don't know the details of your meetings and your ventilation and your spacing, and I don't know the members of your community, it's hard to answer that. And I think that those are things you might want to think about.
Okay, I'm going to try and quickly go through the chat window, and then we'll really open it up for people. So one person asked, can COVID-19 cause diabetes? She's heard that it does.
There's data that has come out about that within the past week or so. I think that people are still examining that. It seems like this virus changes the regulation of a lot of different systems of your body. It sort of changes a lot of different types of regulation. Given that some of those other things I've seen, particularly some of the changes in the blood vessels, the idea that it could change hormones and lead to diabetes doesn't shock me. But it's still very early days in understanding the data, because those data only came out this week. And so I think that that's something that we need to keep an eye on. I wouldn't be completely shocked based on some other things I've seen, but we do need a bit more data on that.
Okay, then we have a question, is the Omicron variant itself a variant of a prior coronavirus? And I think he means non-COVID related.
Yes. So it's sort of an interesting question, because about a year ago, we were starting to talk about what was then called the UK variant. It's now called Alpha. And some people were calling it a strain. And instead, we really got into this word variant. And that's because strain has some special meanings in virology, and virologists were arguing whether it was really a strain or not. So we picked a neutral word, variant, which doesn't actually have a really specific definition. Yes, COVID-19 is caused by a coronavirus, SARS-CoV-2. It is another coronavirus, and we can make what's known as a phylogenetic tree, sort of like a family tree of all of those coronaviruses. It looks like SARS-CoV-2 branched off of that tree from some of those other viruses about 50 years ago, and has been reproducing in nature for about 50 years. So in that sense, sure, I could say it's a variant of a prior coronavirus, but not one that we were familiar with before, sort of the 50-year-old ancestor of one.
Another question, how does the virus affect unborn babies? And I guess this person means if the mother gets it. And a related question, how does it affect small children, even infants?
Yes. So there has been some data showing that women who have COVID-19 during pregnancy can have some negative pregnancy outcomes. And so unborn babies can be harmed by their mom having COVID. And this makes it incredibly important for women to be vaccinated if possible before pregnancy so that the babies are protected. We haven't seen any issues with vaccines. If you've heard about them, a particular issue with vaccines, that issue actually was something that somebody came up with as an idea in a blog post and doesn't have any science behind it. And there are a number of cases of small children who have had negative outcomes. They are less frequent than in older individuals, but they do still happen. And I believe it is 40% of children have other comorbidities, but there are still also children who are healthy children who sometimes will end up sick due to COVID.
If you're told you're positive,
Mhm.
COVID, are you positive to the variant or to the virus itself, or is there a difference?
We can't really know that for most of the tests that we use. There are absolutely tests that can be used in a research lab to tell the difference. And there is one test that is one kind of PCR test that sometimes can tell the difference. But most of the tests we're using on a day-to-day basis just test for the virus in general and don't tell us which variant of it we have. They're just looking specifically for SARS-CoV-2. Sometimes we will do the sequencing in the laboratory to figure out which variant it is, but that is not done routinely.
Right now, today, is it safe to meet with vaccinated friends indoors with masks? Should we be double masking? And when traveling, should we also wear a face shield in addition?
So the face shield is the easiest part of that to answer. So face shields have not seemed to be particularly useful in kind of settings outside of healthcare settings unless you are trying to protect your eyes. So the airflow that's coming around a face shield is not sort of going to do much for you day to day unless you are, say, a healthcare worker who may have a splatter and coming towards their face. And so I would not worry about a face shield in sort of normal daily life unless you have a particular worry about your eyes, although I have not seen a lot of transmission in eyes. And so a mask, particularly a good N95, KN95, or KF94, tends to be rather sufficient. Then I have been around vaccinated friends and family members indoors in masked situations. I know in those situations, I think about whether or not I am around individuals who are particularly immune compromised. And so there are some individuals who I will be around without a mask, others who I will not because I worry about their immune systems. I also think about sort of the length of time, the ventilation in the area. So when I'm in my laboratory, I'm never around anyone without a mask because we have pretty poor ventilation, as well as the distance that I'm spending from those people. And so I take all of those things into consideration, which make it a little difficult to answer since I don't know you or who you are planning to meet with.
And speaking of masks, are N95 masks really safer than, say, cloth masks? And a related question from someone who's heard that you're not supposed to put a cloth mask over an N95, but she says that thinks she gets a better fit by doing that.
So the data that I have seen, and there was some data that came out relatively recently on comparisons, did indicate that N95, KN95, and KF94 did give you better protection than cloth masks. I think that I have seen recommendations on cloth masks over KN95. So I think that especially if you feel like you're getting a better fit, that should be okay. But there is specific data on the other types. I believe on the CDC's website, you can actually get some specific information on the other four, or on the other three, sorry.
Are serology tests worthwhile? And why don't you explain what a serology test is?
Sure. So serology tests are tests that are looking for antibodies in your blood to see if you have made an immune response against SARS-CoV-2. So we can look in your body to see if you have antibodies. One thing that's sort of interesting about a serology test is we can test antibodies that bind to different targets. So we could look to see if you have antibodies against spike, the lollipop protein you saw before. Or we could look to see if you have antibodies against a different protein in the virus, N. The reason why I mention this is that you only have antibodies against N if you've been infected. You can't get those from the vaccine because the vaccine doesn't have N in it. And so you can actually tell the difference between an immune response that came from infection and one that came from vaccination. That also means that you should be careful if you're trying to get a serology test to see if your vaccine worked. Because if you get the wrong kind of antibody test, you're going to be testing for antibodies that didn't come from vaccines. And so that test shouldn't test positive. I've had a few people write me emails about this, and that's often been the answer. Serology tests can be useful in telling you whether or not you've made an antibody response. However, the problem is that I don't know, when I look at data from a serology test right now, what number of antibodies is a good number. I don't know if you have 53 antibodies, if that's enough to protect you or not. We don't have a bar there. So we can tell you, yes, you have some or not, or no, you don't have any, but we haven't done the experiments yet to really let you know what some of those numbers mean. What is really good, what is not really good. And so it can be, give you some information, but I would not say based on a serology test whether I was certainly protected or not, because we just don't know the answer to that. It might be that the thing that protects you is T cells, and T cells aren't measured in a serology assay. So we just don't have that piece of the puzzle yet.
I have a two-part question. Can vaccinated people spread the virus? That's part A. And part two, if you've already developed an adaptive immune response from past exposure, can you, from that, spread the virus?
So in both cases, the answer is yes. I mentioned before that you could be infected. And when you are infected, you could potentially spread that virus. The data that I've seen makes it look like you might be able to transmit a little bit less well, or maybe for fewer days. So maybe a person who's unvaccinated has enough virus in their body to transmit for 5 days, and a vaccinated person only has enough virus to transmit for 1 day. And I'm making those days up, but those aren't, don't quote me on those days. And so, but you could potentially spread, and that's true whether you've been infected or whether you've been vaccinated. You do have the ability to potentially spread to others if you are infected.
I think the person may have been asking if you have an adaptive immune response.
Mm-hmm.
And I guess maybe he's asking if you're no longer sick. Can your immune response spread the?
Oh no, the only thing that will spread is if you have the virus in your body. If you have the virus in your body, the virus can spread to another person. Once the virus is gone from your body, you are no longer able to transmit. The immune response does not transmit.
Okay, and which is better? Which gives you better protection? Actually, having COVID or being vaccinated?
with there are some people who get COVID who make very good immune responses. There are also some people who get COVID who don't make very good immune responses. There's a big range in what the immune response looks like in people who are infected. And people who are vaccinated, we don't see the same kind of range. Everybody's up at the top making a very good immune response, whereas the people who are infected have a bit of a range. In addition, and so in that sense, there are some people who get infected who have good responses and some who have bad, but everybody who got vaccinated has a good response. In addition, when you get infected, you get all the parts of the virus in your body. Some of those parts seem to have the job of messing with your immune system as part of kind of a virus versus immune system battle. And so when you get vaccinated, you don't have all those other side parts that are messing with your immune system. Whereas when you're getting infected, you have things that you're making an immune response to, but you also have some of those other parts of the virus that are trying to mess with your immune system at the same time. So from that, I would say that individuals who were vaccinated have particularly good immune responses. There's one study that actually looked at people who were infected and then got 2 doses of an mRNA vaccine. And they had the best immune responses I've ever seen to anything. So if you have been infected and you can still get vaccinated, it will make your immune response better, and it will make your immune response kind of like superhero immune response. I've never seen anything like it.
What about the reverse? If you're vaccinated and then you get it?
I haven't seen those data. And so I don't know if they come out quite as good. I might expect reasons why they might, but I haven't seen it. So I cannot be quoted on that.
Okay. Can the virus, we'll take one more question from the chat window. Boy, we had a lot. And then we'll open it up. So people, if you want to ask your question in person, so to speak, think about it and raise your hand virtually. Can the virus be transmitted by touching something or someone who has the virus? How is it transmitted?
So viruses are only able to reproduce when they are around our cells. They're in cells. They can dry out and be inactivated pretty easily. So what you have to do is you have to get this virus into some living cells in your body, either cells in your mouth or cells in your nose. If you got the virus on a surface and it sat there for hours, it would dry out and it wouldn't be able to transmit. So if it was on that surface for a very long time and you touched your nose, that wouldn't be enough to transmit. Your hands, the top layer of your hands are all dead cells. The virus can't get into your skin. It has to get in through nose or mouth. If you happen to touch something, say I sneezed on my desk and you touched my desk right afterwards when the virus hadn't had time to dry out too much, and then you touched your nose, maybe you could get some spread that way. But it's really something that we think about with sort of going through the air to your nose or sort of droplets that way. So hand washing and hand hygiene before touching your face is really the biggest thing that will be important for spread by touch. Of course, there are other things that can be spread. You should still wash your hands anyway.
Okay, Richard Askins, please unmute yourself and ask your question.
Thank you. This has been very informative. I am immunocompromised because I had a bone marrow transplant in 2018. It hasn't fully come back yet because I had leukemia. You mentioned that they don't know yet for immune compromised people what the effect of vaccination is totally. I'm wondering if you would be willing to venture an estimate or a guess as to how long it might take to do the necessary parts of the research to determine that.
So that's a really interesting question. I hope that you're doing well after your leukemia.
Thank you, pretty well.
Good, I'm glad to hear that. So it's an interesting question. So you mentioned immunocompromised individuals, and the reason why that's really hard is that there are lots of different ways one could be immunocompromised. And so in order to really answer that question, one would have to look at people specifically with some of those different types of immunocompromising conditions. My recollection is that the data is out there for patients who have gotten transplants. I don't remember it off the top of my head, but I believe
okay.
that one has actually been done and has been published. It's just not for immunocompromised people as a whole. But I think that this specific type of immunocompromise has actually been looked at. And so I think there are studies out there.
Okay, thank you very much. So it's kind of indefinite. That's okay. They keep me fully vaccinated, no problem.
Good, good.
Okay, we have a
Thank you.
question from Chris. I don't know your last name. Chris, if you'd unmute yourself and go ahead.
Hi, Chris Sentner. I'm in Northern Virginia. Could you, by chance, briefly go over the issue of what an endemic is? And it's my understanding that this disease is going to become endemic, like the cold or flu. And I was wondering if you would briefly discuss this topic. Thank you.
Sure. So there are a couple of different patterns of disease that we talk about epidemiologically. One of them is pandemic, which is a word you've heard about a lot lately. One of them is epidemic, and one of them is endemic. Something that's endemic circulates at sort of a background level at all times. So, for example, I often tell my students that in the area of New Jersey where we live, Lyme disease is endemic. It's always there's always some possibility of getting Lyme disease. It's always around in the world. And what we think will happen is that this virus will become endemic. So we're not going to get to a point where we eradicate this virus from the whole planet. We're going to always have it circulating through the human population at some low level. There are other coronaviruses. There are 4 of them, in fact, that are already like this in the human population. They cause common colds. And they're always around at a certain level. In fact, there are some people who are unlucky and get a cold in July. And there are others who, will get a cold during the winter season. Usually we get these viruses sometime as a kid, runny nose number 17 when you're 3. And then we get some immunity and we are protected for the rest of our lives. We don't get sick because of that immune response, even if we might get infected. And so we kind of think that this virus will eventually look like a 5th common cold virus in terms of our interaction with people. What you notice is that if you think about those common cold viruses, it's very rare for someone to get a common cold for the first time when they are older. If they've had to, live in a bubble their whole life, to not get a cold until they're older. And so it seems as though if you get one of these infections as a child, it tends to be less serious. You get some immunity that protects you long term. And so we think that that is what is going to happen. Part of that is just because of the number of people in whom this virus is right now. We've never been able to successfully get rid of a virus that has made it this far around the world and into this many people. We've only managed to get rid of 2 viruses ever, to our knowledge. And those took a fair amount of work and had a fair amount of sort of special things in their favor. And we also know that there are a number of animals that are getting infected with this virus. And so even if we somehow manage to treat every single person, those animals would still have it and could potentially spread it back to people in the future. So this virus is going to be with us forever. And we have to think about kind of how that is going to look and what kinds of things we're going to do in order to help us once we get the virus to sort of a lower level. And so eventually we'll get to numbers of cases that are similar to numbers of cases and severity of cases of colds, but we're not there yet. And right now we need to protect the population and protect the healthcare system until we get to that point.
Question from, iPad 5, whoever you are, would you unmute yourself and ask your question? And I'm going to ask that person. Okay.
Hi, it's Lucy. I'm iPad 5. My battery died on my laptop, so I had to transition to another device. Are we likely, do you think, to be seeing another booster shot, so a fourth vaccine, get recommended within the next, say, 3-4 months?
I have not seen any data on another booster shot. And I think that right now, I don't expect to see that because I have not seen any the data on that. So the companies have not yet started doing any studies of that. And so before there would be a recommendation, they would have to do some studies. For some reason, there's some question about whether Israel is going to start recommending it on 0 data. And I'm not so sure about that. But
yes, I know someone in Israel who did get a fourth vaccine. Yes.
Yeah. Yes. And I am aware of some people who have as well. The FDA would not approve it without data. And since the companies have not yet started any kind of trials on that, I don't imagine that we could see that kind of recommendation anytime soon. I would need to see a fair amount of data before I would be convinced that it would be necessary. Right now, I think that we're probably okay with the vaccine doses that we have.
Thank you.
Yes.
Eric Sandhusen, unmute yourself and ask your question.
Great, thank you. And this has been very interesting. You did allude a little bit to one of my questions, but I'm not sure I really, and maybe there isn't an answer, but as I understand it, right, so the rapid tests are, they're looking to see if there is the spike protein, or is that right? They're not looking for the antigen, they're looking for the spike protein?
No, they're looking for an antigen, which is one of the proteins from the virus.
Okay, so I guess my question is, are all of these, the rapid tests that we're doing now, they're specific to SARS-CoV-2, or is it possible that they're also identifying the earlier coronaviruses?
So they are looking specifically at SARS-CoV-2. When the companies generated them, they had the other earlier coronaviruses in the labs and actually specifically tested to show that those rapid tests would give you a positive only for SARS-CoV-2.
I want to ask a follow-on question about the rapid test that Eric mentioned. So a couple of days ago, the New York Times had an article which said that there were a couple of studies out that suggested that the rapid antigen test may not pick up the Omicron variant as well as the other variants. Do you know about that? And should we be
there have been some examinations done. I think my understanding is that it depends on which rapid test you're talking about, which brand you're talking about. When I looked at those data, I have tended to use the rapid test from Abbott. I've been using one called Binax now. It's just the one that I've been able to get my hands on. And I remember reading that study and noting that Binax was fine at detecting Omicron. It was sort of one of the top one at detecting Omicron. Omicron didn't matter. And I honestly didn't read much more of the study other than to say, oh, the ones I have are going to be fine. I need to go back and look at that study, but it was a brand to brand issue.
Okay, Mark Hecht, please unmute yourself and ask your question.
Okay, just 2 very brief questions. One is, if the vaccine is so harmless, and what would be any reason not to give a 4th booster? I mean, there's no downside to it, according to what most of the media would suggest, or it's such a boon that what would be the downside of taking a 4th booster, and wouldn't it? So how could it not be the best thing to do if it's? I don't see what the reservations would be. And also another hypothetical question: if you had to go into a room or a meeting with people who had all been vaccinated, but there was no quick test before the meeting, or a meeting where none of the people had been vaccinated, but they had a quick test, So everybody before the meeting would take the test before the group. Which group would you feel safer in? The group had been vaccinated and where nobody took the test, or the group where no one was vaccinated and everybody had to take that quick test? So two separate, yeah.
Sure. So the first question is a pretty straightforward question, actually. So the biggest reason why we wouldn't recommend right now that we give fourth vaccine doses is that there is a tremendous issue around the world of vaccine equity. And so while we have enough doses, I think, in the United States to vaccinate everyone like 10 times, there are countries around the world where far fewer than 10% of the people are getting vaccinated. And that means that they are getting infected. That means that viral replication is happening there. That is not great from a health and equity perspective. It also means that the virus has more chances to mutate and make more variants that can spread around the world. And so we need to make sure that we are giving those vaccine doses that we have to some other countries. There are countries where their doctors are not vaccinated. I do not see a great reason for us to be getting a fourth vaccine dose before those doctors get there first. And so one thing that we absolutely know is that, first and second doses are certainly more important than fourth and so on, or even third doses in a lot of cases. And so the biggest reason I would say, as an individual, there's not a great reason not to get a fourth vaccine dose, but as the world, as a population, those vaccine doses can be better used elsewhere. In terms of the question about which room I would want to be in, that is a tricky question. I'm imagining these two rooms and I think that it's hard because when I imagine these two rooms, I'm thinking about people I know who are vaccinated and unvaccinated, and the two rooms I'm imagining happen to be different rooms in terms of how many people are in them and things like that, given just who I'm imagining as unvaccinated people who I know. I think that I would probably want to be in the room of vaccinated individuals myself. And the reason why I would want to be in the room of vaccinated individuals myself is because that way I would know if I had had a false negative test, I would be unlikely to spread to someone else and really lead to them being severely ill. Whereas if I was in a room with unvaccinated individuals, and I happen to be positive and not know it, I could lead to some pretty severe disease in someone else. And I don't know if I could actually go to sleep at night having known that I did that. So I think that's my answer, but I agree with you. That is a challenging question.
Yeah, but the thing is, you suggested that the vaccinated people could spread the disease if they can get it and spread it.
Right, but you're less likely.
Okay, we'll take 2 more questions. Sue Akiska and then Javier Valencia. Sue, if you'd unmute yourself and ask your question.
Yes. Now that we have the Omicron variant,
It's so good to hear your voice.
Whoops.
does that mean that Delta is no longer here and the prior variants are no longer here, or are we potentially getting infected by any number of variants?
Right now, you are potentially getting infected by any number of variants. And so from the data that we have, Delta is still here, as are some of the other variants.
Javier, please unmute yourself and go ahead.
I wanted to know, I just got the virus and I'm vaccinated. How likely is for me to be intimate with somebody and get him sick?
So you may spread the virus to someone else. it is possible, particularly if you are, say, during the sort of 1 day-ish period where the virus is at a high level. But you asked the question of how likely you are to get them sick, and that depends on some things about them. That depends on whether or not they are vaccinated, and some details of their health. So it sort of is a question of who is this other person with whom you are having contact.
So my question is still a little bit foggy. I just got the virus. I went 10 days already from the virus, and I want to go. How is it safe for me to be around people? And now that I got the virus and I'm vaccinated, how long does my immune system stay stronger than others when I got the disease?
The data I've seen had those immune responses looking pretty strong for a month or two. I don't know if we've looked to see exactly how long they stay up, because we just didn't look long enough. The people didn't stay in the study for a very long time. So it certainly seems to be pretty high for a month. It could be longer than a month. So your immune response still should be in pretty good shape. The CDC guidelines say that if you are vaccinated and got an infection after 10 days, the CDC guidelines say that you could be around others, though I think a lot of people like if you have a rapid test just to show that you are not transmitting.
Okay, now I promised to get Dr. Barker out of here in an hour, and we're already 9 minutes over. So I don't know if you've got a minute to talk about women in science.
Sure.
Okay, you're free to go if you want. I really apologize for keeping you. I know that's an interest of yours. I looked up some statistics about graduating classes in medicine and in law. And in those two fields, women are at least as many of, when you look at current graduating classes in medical schools and law schools, women are 50%, sometimes a little above. What about the science and technology and math fields? And if it's different, why do you think it's different?
So there are big differences within the sciences and also within different scientific fields. So the number in general will be lower within the sciences, but it actually is closer to 50-50 in some of the biological sciences. In, say, physics, it's actually quite far away. So there's a big range throughout the sciences. I think that some of that has to do with sort of the challenges in the some of the challenges in the day-to-day of being a scientist can be difficult with trying to manage some other aspects of life, but that doesn't get at the graduation rates that you mentioned. I think that, women and girls are generally not encouraged to be good at science. And one thing that I often find, one thing that's actually very important to me and why I like to do things like this, is that people have a stereotypical idea of what a scientist is and who a scientist is. There's a study that I often think about and I often talk about called the Draw a Scientist Project, where children were asked to draw a picture of a scientist. And when that was first done, if I remember correctly, it was 97+% of the children drew a man. The numbers have gotten somewhat better now, but there is sort of this idea of who a scientist is. In fact, if I had been asked to draw that as a child, I would have had a male scientist who had messy hair and stains on his lab coat and looked a little crazy. There's sort of this idea of who a scientist is. And I think that there are a lot of people, women, but many others, who look at that idea and say, that's not me, so science must not be for me. And they go to work on other things. And I think that it's very important for people to realize that scientists are just like people, like everyone else. There's a sort of thinking process that we are trained to use that we spend a lot of time with, but anybody can be a scientist. And even my college students, I feel like, get this, and we have a lot of women in our science departments, and so they see this, and I've taken them to conferences with me, and they've walked around the conference and they've been like, wait a minute, really every kind of person is here. There really is every kind of person as a scientist. And they were students who I would have said, oh, they definitely, don't have this preconception already. And so I think that just making sure that we show girls and we show people in general who scientists are and what skills it takes to become a scientist will really help that out. People think of scientists as being, good memorizers and just having lists of facts memorized. But really the skills that I use the most often are my creativity and critical thinking skills to figure out how to solve problems when different crazy things happen in my lab. And I think that we need to make sure people understand that those are the skills and those are the things that we really need in our scientists, and show everybody kind of that a scientist is just, you're a person like all the rest of us.
And one other question on this topic: what about obstacles in terms of the career path for women? And I'm talking especially about child rearing, because we know that whatever people might say, the child rearing tasks tend to fall to the woman. I know many people also have a picture of academics as having this cushy job. They teach 1 or 2 or maybe 3 classes. They go home. They've got a lot of time. So what about that?
Yeah, so I teach 3 classes a semester, but I also run a research lab, mentor students, am on a whole lot of committees. I would love to have the free time that people think I do. That would be really fun. Maybe I get enough sleep. But there are lots of ways that sometimes science to me has been really flexible, but I know that has been because I've had some really good colleagues and really good mentors who've helped make sure that I've been able to have some of that flexibility. And it's not easy. Some of the years where in theory, as a scientist, you should be kind of in the lab at the bench working really hard are years that women tend to want to be having children. And so how you kind of balance all of that is a challenge, and it's a challenge for everyone. And it depends on details of your partner and your boss and your colleagues and your institution and sort of how your family and what your support system looks like. And I know people who've done an amazing job, and I know people who've really struggled with it. And I think that making sure that we have more options for women to be able to have a support system that makes this work, and increasing that flexibility would help us in general at having more representation of women in science.
Okay, well, we've kept you 15 minutes later than I promised. I apologize, but I'm really glad you were able to stay. Dr. Barker, I cannot thank you enough for joining us this afternoon. You really answered a lot of people's questions. We have a lot of great comments in the chat window. Thank you so very much. And I look forward to the next episode of TWIV.
Thank you so much.
Okay. To learn more about us, visit our website, ethicalfocus.org, or email us at admin@ethicalfocus.org, and we'll get back to you. To make a donation, go to ethicalfocus.org/donate. Please follow us on Instagram, Twitter, and Facebook, and you can watch many of our past programs on our YouTube channel.